Carbohydrates, they're a type of molecule that we should all have heard about before. So stuff like starch or sugar, those are carbohydrates. Okay, what else? Well, actually, you notice on nutrition labels that dietary fiber is listed under carbohydrates. Why? Because it's mostly cellulose. Dietary fiber is mostly cellulose, so cellulose is a string of glucose molecules that have been linked together with beta glycosidic linkages. Okay, now what about lipids? Lipids, well, they include fats, they include cholesterol, and they include some other stuff like waxes, but those aren't exactly shown on nutrition labels. Okay, carbohydrates, well, they have the empirical formula of CH2O. Now, mileage may vary depending on region on whether you use the round or the square brackets, but usually they tend to be round. Why? Because square brackets, generally in chemistry, that's used for concentrations. Okay, well, monosaccharides are simplest forms of carbohydrates. They actually keep to this empirical formula, which is very important. So they have a single sugar unit. This is a building block for more complex carbohydrates. So you can encounter them in various isomers. For example, ribose as a pentose sugar, so it has 5 carbons, may have isomers that are, say, straight chains. But wait a second. If we recognize this and this both as glucose, what does it take to actually make an isomer? Well, you have to switch around the orientation. Now, there are rules when you draw these diagrams that govern, hey, this indicates that the OH sticks forward. This indicates that the H sticks forward, slightly out of the page. So what does this actually show? Does this actually show a straight chain of carbons? No, it actually shows a pretty much almost a ring already of carbons. So it bends, and all of the groups are shown coming out of that ring. This is very important because, well, isomerization in this case basically depends upon the stereocenters. Okay, so there can be alpha glucose and beta glucose. Please don't call them A and B glucose, even though it's remarkably common and people will know what you're talking about. What about distinguishing between, say, fructose and glucose? Well then, as we can see, fructose has, okay, it has a different ring pattern, a 5-membered ring, and glucose is a 6-membered ring. So glucose and fructose are both hexose sugars because they have 6 carbons. But okay, how do we actually distinguish them? Well, when we draw them out, fructose ends up being a ketose sugar. So in other words, wait a second. When this bond forms, by the way, the specific mechanism is not as depicted here. The actual mechanism is that the oxygen takes one of its lone pairs of electrons and attacks the carbon. And the carbon, well, this oxygen picks up a negative charge. So what happens next? Well, passing water molecule picks up the H+, so that the oxygen here is no longer positive. And then another passing water molecule gives this an H+, so that the oxygen there is no longer negative. And you can end up with, say, this conformation, or because for aldehydes and ketones, you have a double bond, and therefore the whole thing is a flat, instead of what happens when you have 4 single bonds on a carbon, where you end up with tetrahedral, i.e., oh, this, okay, and this sticks out of the page, and that goes into the page. Yeah, no, when you have a double bond, this is flat. When you have a triple bond, it's all in a straight line. So this can end up being OH on the bottom or OH on the top of this ring. And, well, that has some implications in terms of bonding with other glucose subunits. Oh, wait a second. Hey, what happens if you say purify alpha glucose? Will it stay that way? No, because in solution, in aqueous solution, this is in equilibrium. So in other words, you'll go from this to that, and then you'll go back. And thus, you very quickly end up with roughly equal parts of alpha and beta glucose. However, if you bind them together, whether as disaccharides or just polysaccharides in general, they don't go back. So disaccharides are made from two monosaccharides, and this is called a glycosidic bond when you form a bond between two monosaccharides, and this is formed by condensation or dehydration, depending on preferred terminology. It involves the removal of water, and alpha linkage can be thought of as holding hands. Whereas beta linkage can be thought of as, one person's arm is stuck up like this, and the other person has to figure out how to hold their hand while their hand is stuck down. Well then, what do? Get up on platform next to friend, and there you go. So, what this means is that these two can be seen as, the rings are right next to each other, whereas this, it has to be staggered to accommodate. Of course, the rings aren't actually flat. That would be too simple. And, well, the actual work is a little bit more complex, but then again, that's why biology students kind of have to take chemistry. Okay, on the subject of polysaccharides, a complex carbohydrate can have hundreds to thousands of monosaccharides linked together. They are essential as part of nutrition and a valuable energy source. A human being is supposed to derive somewhere between 55% to 75, 85-ish% of their daily energy intake from carbohydrates. That's considered healthy. Of course, any more than that, and it gets a little bit problematic because are you sure you're getting enough protein in your diet? But regardless, carbohydrates, such as in cereal grains or tubers like potatoes, are an essential part of the human diet. So a polysaccharide molecule has many linked monosaccharides. Polymerization is a process where monomers, which are small subunits, join together to make a polymer, which is a large molecule that is made from monomers. So when you decide to join sugar subunits, usually they're stored as glucose, even though glycolysis converts it to fructose before actually proceeding. So with glucose, well, they can be stored as amylose, which is basically one long chain. Amylose is considered the archetypical starch, such that starch-digesting proteins, enzymes, are known as amylase. To amylase. By contrast, lactose would take lactase to digest. And nucleases digest nucleic acids. Okay, so here we have branching. We actually have a lot of branching for glycogen. Because, hey, we have these hydroxyls just sticking out here. Why don't we put them to use? Now, what happens if you branch, unlike amylose, but don't branch as much as glycogen? You get amylopectin. Amylopectin. Okay, so what happens if instead of these alpha glycosidic linkages, or alpha 1,4 glycosidic linkages, you see beta 1,4 glycosidic linkages instead. Well, those they can't be digested by amylase due to the specificity of the enzyme. So you'd be dealing with cellulose, and each molecule itself is not very strong, but when you bundle it together into cellulose microfibrils, they're used for plant cell walls, and they're important for dietary fiber, so irritate the intestinal lining into producing mucus to ensure regular defecation and prevent complications. They are also used for many other purposes, such as a paper you may or may not be writing on right now, and the clothes that you are probably wearing. Of course, it might be the case that you're not wearing cellulose-based clothing, if it's, say, polyester. Now what happens when we add some side chains, so functional groups? Well, we might get stuff like, say, chitin. Chitin or chitin, depending on pronunciation, potato, potato, tomato, tomato, whatever. Well, it's used in fungal cell walls. It's also used in arthropod and crustacean exoskeletons. It is fairly strong. It's used in human applications for biodegradable thread for stitching wounds. Why? Well, the human body is capable of breaking it down over time. Now, chitin is pretty unusual in that it has nitrogen in its functional group. Wait a second. Biologically available nitrogen is pretty expensive. That's why nitrogen is a pretty much essential part of fertilizers. Well, anyhow, in general, we can have this table summarize for us the structures and functions of carbohydrates. So, monosaccharides can be in chains, alpha rings, or beta rings. They function as energy sources and monomers, so building blocks. For example, there's glucose, ribose, and deoxyribose, which has one less oxygen. Disaccharides have two monomer subunits with alpha or beta linkage. They function as energy sources, such as sucrose, which is glucose and fructose, maltose, which is two glucoses, and lactose, so glucose and galactose. Okay, now polysaccharides are very long chains, or they can be branching chains to varying degrees. They can have alpha or beta linkages. However, they tend to be uniform within a particular polysaccharide. So they function as energy storage, structural support, and sometimes as cell-to-cell communication. So examples of polysaccharides, generally people talk about starch in any of its many forms, and cellulose, which is the most abundant biological molecule on Earth. Now, what's the most abundant molecule in animals? It is the collagens. Now, collagens are a whole family of proteins. They are not nearly as uniform as cellulose. And what else do we have to consider that makes collagens lose out to cellulose in a major way when it comes to abundance? Guys, there's going to be more biomass of producers overall than consumers in most ecosystems, especially if the producers don't have a ridiculously fast turnover rate. Why do we know this? Well, for phytoplankton, you can actually sustain a larger population and larger biomass of zooplankton on a certain biomass of phytoplankton if the phytoplankton reproduces fast enough. But the energy, well, the energy always shrinks as you go up trophic levels. So go up the food chain, you get less and less energy. Now, lipids. What does lipids have to do with energy? Did we just suddenly change topics? Not quite. So fats, So triglycerides that are solid at room temperature, and oils, so triglycerides that are liquid at room temperature due to unsaturation, well, they are lipids and they are very high energy. You will find oils in plants when mass or volume really matters. Otherwise, plants will store their energy as starch. So in other words, what does this actually say? Hey, when does a plant have to move? Animals store their energy in the long term as fats because they have to move around and carry their energy stores with them. So, what about plants? When do they have to move? Well, they have to disperse their seeds or spores. Certain spores of seedless plants are actually so rich in oils that they used to be ignited for camera flashes. That was how oil rich they were. And those weren't even seeds, they were spores. Now, seed plants, well, I'm sure we are all familiar with stuff like, oh, sunflower oil or almond oil. Those are from seeds. So lipids include fatty acids, fats or triglycerides, phospholipids, steroids, and waxes. Fatty acids have a carboxyl group and a hydrocarbon chain of, well, variable length, depending on which fatty acid you're dealing with. There are saturated fatty acids, which are nice and straight, and what happens when you have a fat that has mostly saturated fatty acids? Oh, it packs in nice and neat, and there are lots of van der Waals forces holding the chains together. Guess what? That's going to be solid at room temperature. It'll have a higher melting point because there are more intermolecular forces holding it together. Of course, the shorter the chains, well, they don't tangle together as much. They aren't in as consistent contact, and therefore your melting point will decrease with shorter chains. As anybody who has smelled, say, vinegar can tell you, compared to, oh, hey guys, I take a sample of butter out of the fridge. I take a bottle of vinegar out of the fridge. I unscrew the lid or open up the wrapper at the same time. You will smell the vinegar first because the ethanoic acid is more volatile. Now, oleic acid is unsaturated. Instead of a nice straight chain, we have a kink in the chain. So we would draw this as, well, something like, say, this. What are trans fats? Trans fats, of course, this is abbreviated on both sides. Trans fats would have this happening. So this is abbreviated on both ends. What's the difference? Well, here it's going to be stiff and pretty straight. It'll be even stiffer and straighter than saturated fatty acids. Whereas here, it'll force the overall chain to bend. And thus, what happens when you try to fit them together? They don't fit as easily. They also don't fit as closely. So there are fewer van der Waals forces, or weaker van der Waals forces, between the adjacent molecules. And thus, they don't hold together as well. And that's why they end up being liquid at room temperature. Or at the very least, they end up having lower melting points. Okay, so triglycerides start with glycerol. Glycerol is not considered a carbohydrate. It doesn't fit the empirical formula we're looking for monosaccharides. There are two extra hydrogens. Glyceraldehyde 3-phosphate, on the other hand, is an essential part in carbohydrate metabolism. Well, it's actually essential in metabolism in general, but glyceraldehyde would be a carbohydrate. Okay, so this is how triglycerides, so the fats and oils we usually talk about, get made. You end up having 3 separate dehydration reactions and join 3 fatty acids to 1 glycerol. And you end up with a triglyceride.